Good afternoon, everyone. This is the group 4, and for today, we will be discussing about understanding gene therapy and people cursed with fixing their genes. So let's tackle first the genetic risks. It refers to the probability of the individual carrying a specific disease-associated mutation or being affected with a specific genetic disorder. Genetic risk to health comes from a variety of sources, and predicting those risks can be much more complicated than simply assessing disease inheritance patterns. The majority of health conditions are not related to a single genetic cause, but result from a combination of inherited and environmental influences. So our topic will highlight risk factors and provide risk assessment tools which can improve healthcare decision making. Because chromosomes and genes provide instructions for our bodies, any changes to these structures have the potential to cause genetic disease and negatively impact health. We will discuss the types of genetic diseases, how these diseases are inherited, and the potential health outcomes. Now let's move on to the genetic factors. It seems to play a role in the development in anxiety disorders in humans. It means that part or all of the risk is passed down from one's parents. Here are some examples of genetic factors: familial Alzheimer's disease, familial FTD, familial prion disease, and genetic counseling.
Good afternoon once again. So the next is the genetic risk assessment tool. So the example of this is a family history. So family history plays a critical role in assessing the risk of inherited medical conditions and single gene disorders. Several methods have been established to obtain family medical histories, including the family history questionnaire or checklist and a pedigree. So a common tool used in general practice is the family history questionnaire or checklist. Having the patient complete the questionnaire at home allows extra time for the patient to contact family members and provide more accurate information. It is recommended that all the patient receive a family history evaluation as a screening tool for the inherited risk. So the certain types of cancers such as breast cancer and colon cancer appear more frequently in some families, as do some adverse birth outcomes. So, family history information should be reviewed and updated regularly. Further evaluation should be considered for positive responses with referral to genetic testing and counseling as needed. The pedigree ideally shows at least three generations and involves the use of standardized symbols which clearly mark individuals affected with a specific diagnosis to allow for easy identification. So it may visibly assist in determining the size of the family and the mode of inheritance of a specific condition, and it may facilitate identification of members and increase risk of developing the condition. It should indicate the age of individuals, if disease, the age and cause of death, and any relevant health history, illnesses, and the age of onset. If genetic testing has been performed on family members, the results should be indicated on the pedigree. So the next is the understanding gene therapy. So gene therapy is a medical approach that treats or prevents diseases by correcting the underlying genetic problem. So gene therapy techniques allow doctors to treat a disorder by altering a person's genetic makeup instead of using drugs or surgery. So, the expression of genetic conditions is not always simple as the amount of DNA or the deoxyribonucleic acid present, or the inheritance patterns of a single gene disorder. There are many factors that determines how one's genotype or genetic makeup correlates to one's phenotype or the physical manifestations. Among these are the penetrance, expressivity, mosaicism, and the inactivation. So, the earliest method of gene therapy, often called gene transfer or gene addition, was developed to first introduce a new gene cells to fight a disease. So the next is introduce a non-faulty copy of a gene to stand in for the altered copy causing disease. So penetrance is a measure of how often a disease genotype correlates to the disease phenotype. So, for example, in a completely penetrant disease, every individual who is genetically positive for the disease would have clinical manifestation of the disease. So, in contrast, for diseases with incomplete penetrance, some individuals who are genetically positive for the disease may be asymptomatic. So, the causes of prenatal or the perinatal diagnosis of a birth abnormality or enzyme shortage are examples of situations of the DNA testing might immediately useful. So it should be mentioned that the evidence gathered by the new technology should be kept private and utilized only by the person whose DNA has been read. To our perspective, a person's DNA should be sequenced and should be protected by the law from being distributed to third parties without the individual's express agreement. So this also applies to physicians who have gotten such information who should have treated as they would any other protected material. So the introduction of foreign genes into organism does not appear to pose a risk. The resultant creatures do not appear to be more capable of reproduction than others. Attempts to reduce human suffering disease treatment with somatic gene therapy does not appear to any physical ethical quandaries. The insertion of genes into the human germline, on the other hand, the diagnostic methods made possibly by genetic engineering are extremely useful and powerful. So first and foremost, it will be quite simple to link the existence of specific DNA or the deoxyribonucleic acid polymorphism to illness and other features. That's all.
Good afternoon once again. I hope you're still doing fine. So for our next topic is all about genetic engineering and gene therapy. So genetic engineering is also called genetic modification. It is a process that uses laboratory based technologies to alter the DNA makeup of an organism. It also includes removing a desired gene from a cell, adding it to bacterial gene structure, and reintroducing it into the bacterial cell. The bacteria may be then synthesize the original animal or plant cell's protein. So for the next one is the gene therapy. Gene therapy is a medical field which focuses on the genetic modifications of cells to produce a therapeutic effect or the treatment of disease by repairing or reconstructing defective genetic material. It involves altering the genes inside your body cells in an effort to treat or stop disease. The next topic will be the disease people cured by fixing their genes. Putting genes into plants and domestic laboratory animals is one thing. There are two types of different procedures that are being debated under this heading and they need to be discussed separately. The first procedure is to insert foreign genetic material into the germline into sperm or eggs where they could be passed on to succeeding generations. The second procedure is to use genetic engineering to insert foreign genes into somatic cells into any cells except sperm or eggs under progenitors is an effort to correct some genetic defect. With that, treating human genetic disease by purposely changing the genes that are passed on to succeeding generations seems potentially dangerous and ethically unsound. It is dangerous precisely because of the problems brought up the very beginning. There is great possibility of some great interaction between the introduced gene and the remainder of the genome. The consequences of this ignorance fall onto succeeding generations on innocent children or fetuses. So that's all for our lesson, which is the understanding gene therapy and people cured by fixing their genes. Thank you. I hope you have learned about our lesson. Have a great day.